use of recombinant viruses is a powerful technology, both to decipher the viral replication mechanisms and to provide development platforms for vaccines. Likewise, generation of good quality viral stock is crucial for every viral study, from the most fundamental research to the applied studies. Rescue of recombinant viruses, optimal harvesting, freezing, and titration of RSV stocks are critical methods for all virological studies. They might be considered traditional, but remain delicate and require specific expertise.
The day before transfection, suspend BSRT75 cells in complete medium at 0.5 million cells per milliliter concentration. Distribute 2 milliliters of the cell suspension per well in a 6-well plate. Incubate the plate at 37 degrees Celsius in 5% carbon dioxide until they reach 80 to 90% confluency the next day. To rescue each virus, first mix thawed reverse genetics vectors in a tube. Then proceed to transfection, following the transfection reagent manufacturer's protocol. Next, add 250 microliters of the reduced serum medium to the mixed vectors. In another tube, dilute 10 microliters of this transfection reagent in 250 microliters of the reduced serum medium. Gently vortex both tubes and wait for 5 minutes. Mix the contents of both tubes and wait for 20 minutes at room temperature. Meanwhile, rinse the BSRT75 cells with 1 milliliter of the reduced serum medium.